What's up, APBio Penguins? And I believe the APsolute RecAppers are actually just called cappers. I'm not really sure. So today we are going to be joined by the APsolute RecAP, and we're going to do a Unit 3 Q&A. So I hope we can find Melanie and we can get this started. Oh, there she is. She should be asking me to join in just a moment. Where'd she go? There she is. View request. Let's let her in. So, hi. Welcome, everyone. I hope everyone's doing well tonight. So if just as a quick reminder, hi, today is Unit 3 Q&A. So if you find that you have any questions during this, just pop them down into that little question box. Hi, welcome, Melanie.
Hi, Tiffany. It's Sunday again. Here we are.
It's Sunday, because we chose Sunday at 8 p.m. because we thought that was a good idea. Me balancing the babies, trying to get them into bed, and you trying to get your lesson plans done.
I know. I was on a work call about an hour ago. Just getting ready for the week. But many of us, I'm sure, joining on are also getting ready for the week, and we appreciate you taking the time to learn about Unit 3.
Of course. Okay, so let's see if we have any questions at this point. We only have one question so far. So I think before we jump into the questions, then let's just do a quick, what is Unit 3? What's all is involved in Unit 3?
So we're going to start with enzymes, which, in fact, if you look at the distribution of material, enzymes are quite a few sections of Unit 3. And then photosynthesis and cellular respiration are like the meatier parts. But we don't want you to get too overwhelmed. We'll walk you through kind of like what you need to know, what you can kind of graze over. And then fitness, which is an interesting piece to this section, but it is included technically in the CED. Fitness.
Yeah. And I know that when I was going through the 2020 practice exams, because all of your teachers, or all of us teachers, have access to 3 practice exams. And when I went through that, it was a 9.3 average question. So there's going to be somewhere in the ballpark of like 9 or 10 questions about this unit.
Yeah, it's meaty for sure.
It's definitely a meaty unit. And students always tend to kind of struggle with this. So real quick, let's get our first question while we wait for some other questions to come in. And then we'll jump into our enzyme stuff. So the first question was, it says endothermic plus exothermic versus endergonic versus exergonic.
I've always used them relatively interchangeably from a conceptual standpoint, although they are different. But endo and exo is really where you're going to want to start your focus in terms of like what those root words mean, because you see endo and exo so many times in biology. Actually, if you can think of another term you should know that has endo or exo in it, pop it into the comment box right now. How do you distinguish it for your students?
Exactly what you're talking about right now with the and I just saw a question jump into the chat. Make sure you're putting the questions into the little question box, because we won't see the chat when we're doing our questions.
No.
Okay, so I do the exact same thing that you were mentioning for us to put into the chat, which was how using that prefix word, and I bring it back to, of course, protein synthesis.
Can you not? I know, right?
And so I talk about, and membrane transport, and how when we bring things in to the cell, that's endocytosis. So that's where we're going to see energy coming into the system. And then exergonic or exocytosis is looking at things going out. So how much like the energy is leaving. How do you do it?
Yeah. Similarly, I mean, I think what we always come back to, like, do we defy the laws of physics when we mention energy transfer? And we have to keep our physicists happy. Endometriosis. Ooh, good word. Yeah. Endo. Great word.
Which isn't this like Women's Month? That's a big topic too. Also making sure that
National Women's Day tomorrow, I believe.
Ooh, that's exciting. Yeah. You should have done this tomorrow.
I know, right? So yeah, I focus on graphs. I'm a huge fan of graphs when it comes to working through endo and exo, thermic or exergonic reactions, looking at where do you land, right? Where's the plateau as compared to your reactants? And we'll come back to that and talk about delta G, I assume, at some point.
Also, the person that I was putting this question in there, they're specifically, I think, asking between the thermic versus the gonic part of it. The thermic is literally just looking at energy, heat. Okay. And so like an
Heat, yeah.
endothermic reaction is looking at that it is absorbing that heat. And an exothermic is releasing heat, which is a chemistry kind of focus. You won't see the thermics when we're in our biology world. And then the gonic is actually looking at energy. And so that's where we see the endergonic and the exergonic. It's specifically looking at how much energy is released and absorbed, which brings us back to that Gibbs free energy. And so since we're already talking about Gibbs, I think it'd be like a good segue to just go ahead and mention Gibbs.
Sure.
So Gibbs is just the free energy. It's the energy that is between the products and the reactants, basically. So you would see it like on a graph, and it's graphed by the amount of free energy. And so if your products have more energy than your reactants do, that means that it had to have absorbed free energy, like because it's bringing that energy in, which would have been an endergonic reaction. Okay. And if your reactants have more energy than your products, then that means it's an exergonic reaction and it's releasing that energy. Okay. If it releases the energy, that means it's spontaneous. TNT, trinitrotoluene, is actually a spontaneous reaction because it's releasing all of that energy. Think about an explosion, right? That's a whole bunch of energy being released out. So it is spontaneous. You put TNT on your desk, it is sooner or later going to explode.
At some point. Don't do that, please.
May not right now, but at some point. And fun fact, when I was in my chem degree, a student accidentally made trinitrotoluene. They didn't know what they had, and they accidentally made it. And they're running down the hallway like, I found something. I was like, oh my God, no.
Oh my goodness.
So much
Well,
fun,
it's exactly what I was talking about when I mentioned that concept plateau, right? So we look at the graphs, where are your reactants, where are your products, where do the plateaus of the graph lie in relationship to each other? Especially when we're going to talk about activation energy. And it should be noted, right? Gibbs free energy will not be affected whether you use an enzyme or not, whether that hump or that hill of your graph is high or low. Gibbs free energy is going to be the same, because the difference, or when you see delta, that's that triangle, refers to the difference. And you'll see that delta in front of lots of concepts, but delta G is one of them. That you see the difference is unchanged, right? Because your reactants and products are not going to be modified just because you use an enzyme. The pathway you took to get there may be different, so that's why it required different amounts of energy. But the reactants and products are unchunged. Tiffany, we never introduced ourselves.
Oh, we really didn't. You know me, I'm all about that biology. I like
Get
to
into
jump
it.
right into it. Okay, so hi, I'm Tiffany Jones from AP Biopenguins. I teach in Georgia. Been doing this about 10 years. Melanie.
I'm Melanie Kingett. I am the podcast author for the APsolute RecAP Biology Edition, and I host a bunch of other subjects, and I teach in Pennsylvania.
Ooh. Also, speaking about, you were talking about that the Gibbs free energy isn't different between enzyme or no enzyme. I've seen the question before in which they tell you what the Gibbs free energy is of a non-catalyzed reaction, and then of a catalyzed reaction. They say, like, if I was to double the enzyme, how does the Gibbs free energy change? And you need to make sure you understand that no matter how much extra energy, how much extra enzyme you add, Gibbs free is going to be the exact same. And my students always get tripped up there. They always double the number.
Yeah, and the other thing that we also I find students fall into is it's always final minus initial. And so it's going to be positive or negative depending upon that plateau difference. Oh my goodness, now I have a face to the voice. Yes, hi, Lauren. I'm Melanie. Thank you for listening to my voice in all the bizarre places people podcast. What's the podcast again? Yeah, the APsolute RecAP Biology Edition. So the podcast you can actually find on any platform. They are free. And they match to the CED completely. So I started writing these episodes last year in February, and we have 73, I think, episodes out right now, something around there. So you'll find something for every topic you want, including 7 or 8 on Unit 3.
Woohoo! Okay, so let's see if we have any more questions in our little box. Okay, so okay, so let's go ahead and just, as we are talking about energy, it says, what is important to know about free energy for the exam? So what would you say is important to know about the free energy stuff?
I would say application, like how can that energy be used? So that if you're given a context in which, energy is available or released or stored, you can make a prediction about what that can be used for or how that might be advantageous to an organism. I mean, this is going to relate really heavily to from my perspective, to some ecology later on. You can't, you got to make the energy, not correct, store the energy in different forms, and use it for different things later. What do you think?
For me, it's all about energy coupling, putting together an
Mm-hmm.
exergonic reaction with an endergonic reaction, and how that is what's providing the energy for all processes to occur. And I think specifically in the CED, it mentions that if there is no energy or there's an absence of this free energy, what is going to be the product? And the product is actually death.
Right.
And it's like straight
Yeah,
in the CED is like, what happens if there's no energy? Death. I was like, oh my gosh.
Mm-hmm.
So it is important to understand that in order for any of our processes to happen in our bodies and in the world, is that there's going to be a overall
constant input, and constant input
yes.
is necessary. Constant input.
And I know that there was a free response question about what happens to the extra energy, because I think I did a free response question yesterday in Marco Learning's little live that I did. And in there, it's like, we calculated efficiency, and we were saying, okay, well, ATP was only this amount of energy, and there should have been this much energy in glucose. What happens to the extra energy? And so it's important to know that that extra energy goes to heat, right? And that's what causes our bodies as endotherms to be warm, is because we go through metabolism. So I think the connection also to free energy is looking at the heat energy. We connect it all into everything.
And thinking about, sometimes when we would perceive us wasting energy, but if you need energy for warmth you're going to shiver, right? You're going to shake, and that's going to, use more, ATP.
Well, yeah, because your muscles are contracting in order for you to shiver. That's what's happening. Is you have all these short little contractions, and every contraction requires ATP. I think it's kind of funny. You just drink some water, because usually I'm the one drinking water throughout the whole thing, and I have no water near me, so I was running around. And now I'm like, hey, the times were like, we literally just switched roles tonight. And if you're wondering why I'm reading all the questions, it's because when I save this for later, students won't see those questions. So I try to read them ahead of when we put them up there. If you're wondering as a viewer why I keep reading them. So Krebs and ETC, what is key to know? So what do you find is key to know about the Krebs cycle? Because we don't have to know all 8 steps, right?
So I have a very traumatic memory of cellular respiration and photosynthesis from college, in which I have these memories of being at a whiteboard and just drawing it from scratch over and over and over again, because that was my level. Yeah, it was really bad. But that was the level of expectation at the time. So whether you're talking, cellular respiration, or whether you're talking photosynthesis, it's all about what goes in, what comes out, right? What are your inputs? What are your outputs? And what happens next? And so I find it useful. I don't like to focus a ton on the numbers. I know they're important, but it's a detail that can be easily swapped around.
The question that they had a couple of years back, they gave you the Krebs cycle and they showed you how many it was. So knowing that number is not the end of the world. It's not an end all be all to know that 3, 2, 1.
Yeah, I think relative quantities. I think in terms of it's kind of where I like my students to be, so they can start to see some trends, especially when I know I'm wandering off the question, but especially when we talk about glycolysis and net ATP, and how does that compare to some things down the line. To me, that's where the emphasis is. When it comes down to ETC, I would like my students to be able to compare them in the mitochondria versus the chloroplast, see the patterns, see, the chemiosmosis, see, why hydrogens are moving, and how does that relate back to membrane structure? Because to me, that's foundational to understanding the bigger piece.
Real quick, because this is coming up in the chat. If you are trying to write questions, writing them in the chat isn't going to help, because we need to actually be able to see them, and they go, I don't know about Melanie's screen, but I see one person at a time. So if someone else writes another question, it goes up. So if you look at the bottom of your screen, it says add comment, and then there's a little camera button, and then there's a little question mark. click on the question mark, and that's how you can submit questions to us, and we can pop them onto the screen like we did with this current question. So click that there. Sorry. Oh no, she's circling. Is she back?
I'm here. I'm
Okay.
here. No, I'm here. My AirPods disconnected. Sorry, I should have given you a warning. My AirPods disconnected, so I didn't want the feedback to keep going because you would have looped through. For those of you just joining us, so Tiffany and I have known each other for almost a full year.
Yes.
We had some very traumatic Instagram experiences in the spring when we were really going like figuring it out.
Our first one?
Oh yeah, it was special. But listen, technology is a friend and a foe, and we're gonna use it the best that we can for all of this.
So you were mentioning about the differences between mitochondria and chloroplast, right, with their ETCs. So like I find it's important for students to know, like in terms of that difference, which way the protons are flowing, because we know with the electron transport chain that number one, the electron transport chain does not make ATP. The actual process of the ETC doesn't make any ATP. It's that chemiosmosis that happens next that does that. Okay, but so it's important for me to tell students which direction those protons flow. So in terms of the mitochondria, those protons are going to flow out into the intermembrane space, the space that's between the outer membrane of the mitochondria and that inner membrane of the cristae. So it's important that they know that it flows out into the IM or intermembrane space. And then for the for photosynthesis, it's going to flow in or pumped in to the thylakoid space, or the space that's on the inside. So I like to kind of tell students that it's going into a small area. And so by going into a small space, that way it builds up that concentration to allow for that potential energy that's needed to fuel the ATP synthesis through the enzyme ATP synthase. Remember, enzymes tell you what they do. ATP synthesis is done by the enzyme ATP synthase. Like, it tells you what it does. It's great.
I like the questions that always come off of that, right? Has to do with pH. So if we're moving hydrogen ions, we are going to be, let's see if we can get a prediction in the chat. So if we are accumulating hydrogen ions, are we going to be increasing the pH or decreasing the pH? Hydrogen ions are accumulating in a space. What's going to happen to our pH? Let's see if we can't get a guess in the chat. Is it going to go high or is it going to go low? And pH, you're going to find, you don't have to understand necessarily or calculate logarithmically specific concentrations like you might have had to in a chemistry class. But you are going to need to apply that and then later on see what that does. Good job, guys. Decrease, awesome. Because we are increasing our hydrogen ion concentration, which is going to make it more acidic.
And so somebody has asked in the chat, and I just happened to notice as I was looking for your answer to come through. Mitochondria and chloroplast, those are organelles that are found inside the cell. And so if you look at endosymbiotic theory, which we talked about in the Unit 2 session, was just that those are actually technically they're believed to be prokaryotes that were engulfed. So somebody responded back that it's a cell. Well, technically, we kind of think of them more as organelles. And
and you're gonna
theory
find a lot in common for them, right? So they both have double membranes. They're both energy transducers, right? They do something with energy. Right. They both have ribosomes. They both have the ability to replicate independently. They both have DNA. And so,
circular DNA.
And they're about the same size as the prokaryote, approximately. So if we think about like, why do they have two membranes? And even that concept of like hydrogen ions moving across the inner membrane of the cristae or into the inner thylakoid membranous space, that has some validity to it as well in terms of supporting that theory, because in a prokaryote, you're going to see that happening across the outer membrane for energy usage. Okay. I'm sorry, we get distracted by what's happening down below. So like I'm always looking down.
And I'm always like, I'm looking at the question, trying to figure out which one I want to put in. And I want to remind everyone that this is Unit 3. So it is enzymes, cell respiration, photosynthesis. So if you're putting other questions in there, I love you and I want to answer your question, but I'm not going to answer your questions about something that's not Unit 3, because we're focusing today on Unit 3.
Let's back up to enzymes then for a second. We haven't talked much about them.
Then go back to enzymes. What do you want to talk about?
Go back to enzymes. Let's talk about what they're made of, shall we?
Sure. I'm going to pop this question out, and I hope I can remember to put them back up after. So enzymes are just proteins. So they're made up of amino acids, and we have already talked about that proteins have those 4 different shapes. I'm sorry, the 4 different levels to folding. You've got the primary structure, which is just that single, basically unraveled, where it's all of these peptide bonds between them. You've got your secondary structure, which is your alpha helix and your beta helix, because of hydrogen bonds between the backbone. So between basically the amine group of one amino acid and then way downstream a carboxyl of the other, they have a hydrogen bond between them. So the alpha helix kind of makes like this, almost like a spiral, like a spiral staircase. And then the beta helix kind of makes like a wave. And then tertiary.
You mean beta sheet or beta sheet?
Oh my God, what did I just say? Yeah, beta sheet.
Beta helix, yeah.
This is why there's 2 of us here.
So in alpha and beta, don't get wrapped up in it too much. You're going to see that it's for directionality, like the way things are turning or twisting or bending, things like that.
Yeah. And the tertiary is our final 3-dimensional structure, which is the ending of the bonds between the R groups. And then quaternary is if you have more than one polypeptide. And so the reason why I mention this is because proteins can be denatured. If you change the pH, if you change the salinity, or if you change the temperature and make it extremely high, then they can denature. And so the same thing applies with our enzymes. And so I've seen that a lot of questions on the exam really focus on what would happen if this happens to the enzyme. And so you want to keep in mind that enzymes are proteins, and it goes back to everything you know about proteins.
And a lot of students will think about like what they've learned about reaction rates and enzymes. They're like, well, shouldn't enzymes work better if it gets warmer? And that's true to a point, right? So if we think about enzymes and how they work with their substrates, right? So substrates are the molecule which fits in the active site of the enzyme. Since proteins have specific shapes, they're going to fit specific shaped substrates. But yeah, substrates will bind to that enzyme more frequently if you warm, if kinetic energy increases, right? The energy of motion, it's more likely to bump into that space. But if you
provides
go
the
beyond
activation energy.
Yep. If you go beyond what I call like the Goldilocks zone, right? Enzymes are like your Goldilocks, not too hot, not too cold, just right. You're going to denature, right? So your bonds are going to start to break and you're going to lose your shape. Protein shape is often called globular because it has very specific dimensions. And when we refer to protein folding or protein changing shape, and sometimes proteins change shape on purpose, we'll get to that later. But you get what's referenced as like a conformational change is one of the expressions you're going to see describing that.
And so sometimes this is reversible. Like sometimes you can reverse that denaturation. A good example of that would be if you have curly hair and you straighten it. So you use heat, like a flat iron, to straighten your hair out. But then you go and you wash your hair, and what happens? It becomes curly again. Right. And so there are some times where they are able to reform. And then there's sometimes like when you crack your egg, think about your egg when you crack it onto a frying pan, it starts out being kind of a colorless gel substance, and it solidifies and becomes white because that protein has basically denatured and there's no going back from that.
Yeah, and what's interesting about that is you can see the property also change, right? So you are no longer the egg white, or when it's clear, right? It's no longer transparent. So if you think of it from just like looking out the blinds at your window, and then you pull the mini blinds, so they used to be horizontal and now they're this way and they're no longer transparent. Something has changed in the shape. Therefore, the functionality has also changed. And structure influencing function is a huge theme in biology, from the molecular level to the cellular level, tissue, organism, so on and so on.
Great.
We go on a lot of tangents, so we can't help it.
But they're good tangents.
Right? So we tell
I think that's why people follow and like to watch us, because we have good stuff and it flows. Someone wrote in the chat, what are R groups? So since we mentioned R groups, just an amino acid has 4 things. So it's a central carbon, and then there is an amine group, which is your NH2. There's a carboxyl group, which are COOH. You have a hydrogen, and then there's this R group. The R group is variable. There are 20 different amino acids, and that R group is what's different between those 20 amino acids. If it's a polar R group, then it's going to face outward towards the polar substance on the outside. If it's a non-polar R group, it'll face inward to hide from that aqueous solution outside of it. If it's charged, it'll face outward. So you probably would just make sure that you understand the way that things fold. The previous questions I've seen about it just mention, hey, Here's this side chain, it is polar. Here's this side chain, it is non-polar. How would the protein form?
Right, and then having an ability to understand how it would interact with other things. So you're talking about facing aqueous environments, right? If we have a transmembrane protein that forms a tunnel or a channel that is ion or solute specific, we would see different environments in different parts of the protein. So that would encourage that functionality as well.
Okay, so speaking about our enzymes, oftentimes we have inhibitors for those enzymes. So someone says, can we talk about competitive versus non-competitive inhibitors? So you want to take it?
Yeah, I mean, I think like so many things, it's intrinsic to the word, right? So if we think about what is it competing for, right? So we have a molecule, it's competing for an enzyme, so we'll say attention. Oh, we have props. We have props.
We're always having props. Come on.
So it's competing for that active site, right? It's competing with another, with the substrate for that active site. And there are moments in which an active site can bind at times to like very similarly shaped molecules. Non-competitive is going to bind to a space outside of that active site. Oh my goodness. So can we get a prop back up? Hold on. We need to identify the parts and pieces here. So enzyme is your hand, I'm assuming, right? The pink highlighter is
non-competitive inhibitor.
Non-competitive inhibitor. There we go.
I can't hold it.
Right. So it's binding to somewhere different. And so we reference that as like allosteric. It's going to be a space on another side that influences the active site. And when I say influences, I mean chemically, which means physically changing its shape. Okay. And the green is a competitive. Now we're competitive.
Yeah. Now I went to the other way. So this is a competitive inhibitor. It's bound to the active site. So now the substrate cannot bind.
Lovely. That was very good. I appreciated the impromptu props. We worked together. Okay. We planned this out really far in advance, guys. Totally. And by that, I mean, we just like wing it.
We do. That's what happens when you've been teaching for a little while. Like you just come up with things on the spot and it either works or it doesn't work.
I will tell you, as someone who is like a really visual person, ironically doing a podcast, which is not visual at all, it's been really important for me to think about how to explain things differently, how to provide practical examples, things that you can literally pick up off your desk and make an example out of, because you aren't always going to be looking at a diagram of the Krebs cycle. Like that's just not always going to be in front of you. And therefore we need to have other tools and ways to work with it. All right, where are we at with our questions? We have
I know I skipped that question, so I was trying to find the question to throw it back up there to make sure that I didn't ignore that student. Because it's a student who I know like always posts a lot of questions. I want to make sure that they get their questions answered. So the person's honestly focusing on remembering all the reactants and the products of photosynthesis and photorespiration, which just so we're all clear, you should not try to memorize.
No.
It's all about application. Okay. And so I always tell my students, what does it start with? What does it end with? Where does it take place? And why is it important? And that's like my big go-to is always make sure you know those.
Right. And so if you can identify like where is carbon dioxide like being formed within the process, I think that's a good.
Anybody want to say where in the process of cellular respiration carbon dioxide is formed?
Yeah, and
where do we form it? And where's water formed?
Water, yes, right. So if you look at the overall equations, whether you're actually talking about glucose or whether you're talking about G3P or whatever you're actually focused on, it's important to know where do all of those big players come in. Obviously, there's going to be steps in the middle because we have to get from point A to point B. But I follow the carbons. Like, where are the carbons going? Where did oxygen doesn't get its big moment until the very last, very last step of cellular respiration.
Good job. Okay, so I'm seeing the answers coming in. Pyruvate oxidation and Krebs cycle is where the carbon dioxide is released. Great job.
Good job.
And anybody want to put in where about the water? Where's the water formed
where's
at?
the water?
And so while you're kind of mentioning about following those carbons, I always tell my students to kind of course, follow those carbons. We start out with glycolysis with 6 carbons, right? And remember our words, right?
Lysis.
Glycose, sugar. Lysis is break, right? So we take our 6 carbons and we break it into 2 3-carbon structures. which is your pyruvate, which then moves into pyruvate oxidation, losing a carbon. So now we have one carbon dioxide was released, leaving us with 2 carbon dioxides in each of our acetyl-CoAs to go in the Krebs cycle. And at the end of the Krebs cycle, you have 0 carbons left, right? So those left as 2 carbon dioxides. So you know that pyruvate oxidation plus Krebs cycle is where all those carbons were released at. And so did anybody answer?
Yeah, we did. We did. We got some oxygen as the final electron. I guess some good answers about oxygen.
Yes. Oxygen is that final electron acceptor, which means that it's going to accept those electrons plus some protons, which will then make that water, which is kind of helps you to remember about photosynthesis.
Yeah.
And what's the function of water in photosynthesis?
Split it and donate electrons.
Break that water molecule
to
donate the electrons so that we'll have electrons to regenerate the reaction center complex that moves into its ETC. So they flow together real well.
I can't do this analogy this year, so I'm teaching in person and we wear masks and shields and all the things. So whenever I start to teach cellular respiration, I always tell my students, put your hand in front of your face, take a deep breath in, deep breath out, and what do you feel, right? So you feel a gas.
You can't do that.
You can't do that now, right? It's the opposite of what you can do. But you do feel gas. You feel a warmth, right? So you feel an energy representation. And you also feel moisture, right? So you feel the water coming out of your lungs because that's where they were made in your cells.
And so I have seen this question before about water, and that they use an isotope of oxygen, oxygen 18, and so they can track the oxygen that's in the water. And they always ask you about, where would you see this? And you always need to make sure you recognize that it's released as oxygen in photosynthesis. And they'll give you the answer choices of everything in the Krebs cycle. They'll give you like PGAP, they give you G3P, and all of which have oxygen. And it's so that you can recognize that it's that all those, the oxygens that are in all of those are coming from the carbon dioxide that went into the Calvin cycle.
Yeah, I had a student fall for that trap. I didn't use isotopes, but and it wasn't AP, it was my freshman, but they just told me carbon dioxide because that's the other gas. And it just linked together in their head. Well, of course, it's coming from the other gas molecule from that perspective.
But the big thing also about remembering about reaction products is about the NADH and the NADPH.
Right.
So how do you differentiate between
the P is for photosynthesis. NADPH is in photosynthesis. NADH is going to be in cellular respiration. Super different. Super similar roles.
We have a comment that was made from another bio teacher. I want to call her Kaufman, but I know she got married. But she says in gaseous oxygen comes from water during photosynthesis. Cyanobacteria produce the first gaseous oxygen on Earth. So that's also connecting to the big scope of everything, right? That we know when oxygen was first came about was because of the fact that we can see the iron oxide layers in the rock, and that's how we know that, okay, well, this must have happened, and that's how we had all of the oxygen that was produced in Earth. That's a great connection. Thank you, Ms. Kaufman.
Round it goes.
Name.
Good job. So we're very fortunate when other teachers pop in here because they can be our chat TAs. Keep it up.
Okay. Let's see, what else do we have? Okay, so during photosynthesis, what does the Calvin cycle use from the light reactions? So these two are going to work very closely together. So we need to make sure that that we do know how the light reaction provides materials to the Calvin cycle. Do you want to take it? You want me to get it?
I mean, yeah, you can go. I just think that I think it's so intrinsic to keep these processes separate. And we like to think of them happening like they are distinct. Locations are different. That's important to know, right? That's true in all the organelles correlations, but they're also happening like mildly simultaneously, right? It's not like light reaction and then pause. Yeah. Cycle and then pause.
Unless you're talking about our CAM plants.
I know.
Because they cannot go through the light reaction. They can't go through them together because of the fact that when they open their stomata, they lose all their water.
So CAM plants aren't explicitly on the exam, but they could be in a question. No. They could be like integrated into a question. They'd have to give you the information about them.
Okay, so let's answer this question.
Sorry.
So what is formed in the light reaction? The light reaction makes ATP, and NADPH. I don't know if you said it, but the P for me, I always remember photosynthesis
Yes.
because that's the only way I can remember, like, hey, this is photosynthesis. I mean, yes, APsolute RecAP is an AP Bio teacher. And so what happens is that those two processes, the ATP and ADPH, go over to the Calvin cycle and they're used over there. And so you use both, you have 9 ATPs and 6 NADPHs. And so you're thinking to yourself, wait a minute, those are not the same number. And so in the light reactions, your teacher may have talked about cyclic electron flow and then linear electron flow. So linear electron flow, when it literally goes from photosystem 2, ETC, photosystem 1, and ADPH.
Yes, they are out of order.
Yes, they are out of order. And then cyclic electron flow is looking at photosystem 1, and then it comes back through that ETC, and it literally just cycles around in circles. So that's how you can end up making more ATP than NADPH. And so because they're not even, you have to have that process. Because it would have been the same situation that happens with fermentation where we've run out of NADP plus. So
what do you do?
Right.
Yeah.
So it's the only way that we can make extra ATP without making NADPH. And so students oftentimes will say to me, well, does it alternate? No, some of the photosystems are pre-designed to be
linear,
and some of them are pre-designed to be cyclic. And so between them all, we end up being perfectly balanced. It's just magic. Just go with it.
And the energy is essentially captured, harnessed, and transferred, right? So we think about the photosystem as like all the bits and pieces moving within that within the thylakoid membrane. The role of all of that, whether linear or cyclic, is to capture, harness, transfer into your electron carriers, right? So now that you have them, you can move on to your next step.
Right. And C4 and CAM plants are technically out of scope. C4 is literally just looking at that they're the venous ones that have like this bundle sheath, and they just separate out where the two processes take place. The light reaction will take place in one spot while the Calvin cycle takes place in another, and it has like one extra step. And CAM plants just take place at different times. Why I say this is because of the fact that what the test developers can do is they can explain to you about this and then ask you some type of application question from that. And so you can connect it to something else. So for example, someone just asked about photorespiration. So photorespiration is when the stomata, those pores that you have in your leaf, those close. And because those close to stop the water from leaving, they actually keep all that oxygen it builds up in there, and you're not bringing any carbon dioxide in. And so photorespiration, the ribisco, which is the enzyme for Calvin cycle, takes up the oxygen instead of taking up carbon dioxide, which is slightly a problem because we're not adding any carbons to our cycle, but we're still letting G3P comes out. So it kind of ruins the whole process. So that's what photorespiration is. It's literally that it's going through cellular respiration and not generating any new glucose.
If you're just want to double check, so what question do you have posted right now? Because I haven't had a question change visually on my screen in quite some time.
Oh, I literally just closed that question. The question was about what are the products from light reactions that went to the Calvin. The question that I just answered, I saw it pop up in the chat, and I just segued into it. I'm sorry, and I was segueing into the C4 CAM because a student asked it, and Kaufman responded. So I was just verbally saying some of the stuff that was happening in the chat for those when we post the video later, when it shows up. And I don't know, I was trying to help make sure.
You're good. I'm making sure it's not glitching for me. That's all.
Yeah. Okay. So let's just kind of talk about exam questions for a second then. Sure. Just kind of segue a little bit. What would tend to be a harder question for this unit? What kind of questions do you think that they could give students on this in the exam?
I think a harder question for this unit is really anything that has the Krebs cycle or the Calvin cycle in great detail, because it can be overwhelming just to see that much data. And, whether FRQ, whether it's a long format multiple choice, I always encourage my students to like jump to the bottom. Like look at the actual question first, because maybe it's very possible like half that information is not needed, and many times not necessary to answer the question. I've seen a lot of students get bogged down in like the new terms. They start underlining and highlighting, and while that might be a strategy that's useful, which you're gonna talk about annotation in a second, I can tell. You know, they've also at the end going, oh, I didn't even need to do that. Like I had the information as well for that. Yeah.
Well, you're mentioning about annotation, so you know, you do know that this year there's 2 different exams, right?
I
For
know.
students, you can either take a paper exam or an electronic exam. And when they release the information to the teachers, what was it, like Thursday maybe? It doesn't look like there's any way to annotate those questions.
Yeah.
So personally, I'm going to tell my students to take the paper exam. What is your feedback? Are you going to try to motivate which way?
It is a school decision, right? So some schools are giving some flexibility to their students and some schools aren't. My school is not. So we are not, because we're 100% in person right now. There will be a few exceptions to that. But I'm a fan of paper tests. Really just that you can show your thinking more without it being as high stress. But I think if you are taking the digital version, it's super important you shift your studying now. Like how you practice questions, how you time yourself out, what you give yourself, how you create your study scenario.
Don't go back to anything when you're practicing your questions.
You won't be able to
press that back button ever.
You won't be able to go back and check your work. So that is a different strategy of taking a test. It can be equally as effective, but it requires you to modify the way that you go through things now. Because the worst thing that could happen is create a false expectation of comfort with reviewing your answers and double and triple checking, and then removing that from you on test day and having that be the first experience where you've had to take a test like that. So I have a personal preference, but I think regardless of which path, I think the students should just take the practice how you will take your test, which will be important.
Okay. So back to the question that was asked about what you think would be on the exam. I think it would be a graph. Maybe they'll probably give you a graph about like maybe pH and about what's happening to the pH, and you know, maybe you could talk about like taking a mitochondria and you put it into this other environment. What would you predict to happen to the pH of the mitochondria? What would you predict to happen if you turned off the light? You know, maybe looking at there was a question before about, oh gosh, I cannot think what the blue solution was, but like literally it went from blue to colorless because it took up the electrons, DPIP. So I could see them maybe taking a question like that, because that's not actually a lab that we do anymore. But it definitely is a fair game activity that could be applied. And just to ensure that everyone knows, the test developers can use any lab that they want. It could be an experiment that we've done in rooms. It could be an experiment that you've never done. But no matter what the experiment is, the test developers have to give you the background information about the experiment that you would then apply your biology knowledge to it. So don't be freaked out if, like, when you look at the question, like, oh, we never did this lab. A lot of the country hasn't done that lab. It's designed in that way that you are just applying the biology, not applying your lab skills. Sorry.
Yeah, and that to that point, yeah, very, very true. I think exposing yourself to as many style of questions as possible within the context. So if your teacher hasn't used AP Classroom much, please ask them to. You can ask for extra questions perhaps, and I think just like a little bit more practice with the style of questions is a good way to go. Another thing about graphing, since you did mention it, in the digital version, there are certain skills they're not going to be able to ask you to do. And so you're going to need to extract from graphs, whereas on the paper test, you may be asked to produce some of that. So that's going to be a differentiation between the two styles of tests.
So someone asked the question, and the only reason I'm popping up there is so that we can the person typed in a couple different times. I think they didn't think their question went through. It says, which protein complexes do NADH and FADH2 transfer their electrons in ETC? That is is too detailed. So you wouldn't really need to know the names of the cytochromes. You don't need to be able to know which coenzymes are in there. Just be aware that NADH and FADH2 transfer the electrons from these other processes to the electron transfer chain, you could know that NADH will put it up on a higher point than FADH2. So more ATP is produced per NADH than FADH2, but I wouldn't need to know, you don't need to know the specific place that those are going to be, where they're going to reduce and where they're going to get oxidized. Sorry.
No, that's fine. You mentioned a question about mitochondria and pH way back when we were talking about examples. So just as like a don't fall in the trap, if they're talking about a plant, it's going to have chloroplast and mitochondria. So especially if they're showing you some graph data with different gases and things like that, just don't forget mitochondria are also present.
So we actually have no more questions that have been posted about cellular respiration. There's a bunch of other stuff in there, but nothing about actual. So we can kind of, let's segue over to digital exam for a second. Someone says, can you elaborate more strategies for our digital test taking? So let me just jump in real fast and you give me your stuff. Okay?
Yeah.
So I would time it. Okay. So since you cannot go back, I would give myself on the long free responses, I would give myself 25 minutes. You're going to have 2 long free responses. So that's 50 minutes right there. And then you have 4 short free responses. So I give myself 10 minutes for each one. And so that gives you your full 90 minutes. How would you determine this? Literally set a timer next to you for 24 minutes and spend your 24 minutes doing your exam. And that way it gives you a minute to finish whatever sentence you're on and press the next button to go to the next question. So I would spend 25 minutes for the 2 longs and 10 minutes for the 4 shorts. For your multiple choice, I would try to stay about a minute to 1.3 minutes for your multiple choice. There's going to be 2 types of questions. You'll have these, basically they're independent questions. They're by themselves, as literally just the question as it is alone. And then you have these things called set questions, in which they're going to give you some long prompt that gives you some information. And then you have, I think, like 3 to 5, maybe 6, it kind of varies, multiple choice questions that correspond to that reading. And so I would spend like kind of maybe like a minute or so with your independent questions. And then I would probably put a little bit more time for those set questions, because you're probably going to have to go back to the reading a couple times. I don't know about your ability to remember what you read, but I have horrible reading comprehension, and I have to keep going back to that prompt so many times.
And you're going to be reading on a screen, which is going to be a different style of reading.
Yes. And so I would give myself a little longer for the set questions, but still kind of averaging about 1 to 1.3 minutes. Kind of definitely watch your time.
And figuring out your time now also might take some practice. So it's not even about, I didn't finish. That's good information, but better information is, if I had more time, would I have known the answer, right? If I kept writing, would I have earned points? And that gives you information, yes or no, and how can you shift? And that can just be done very simply with like, okay, the timer went off. I'm going to keep working and maybe like change my color that I'm typing in, so I can see what I wrote when I had a lot of time and what I wrote when I didn't. And over practice, more and more of the meat and potatoes information, you can get into that a lot of time. But knowing why you ran out of time is useful. Were you actively typing, or were you going through, and I'm saying typing because you're practicing for the digital test. So I refer to it a little bit as like test analytics. You have to know what your pitfalls are. So practicing and not finishing is a piece of it, but getting to that pacing, right? How do you train yourself to get to that pacing is useful as well.
And another idea, because you won't be able to cross things out, ask your teacher for a bubble sheet. Say, hey, can you print me up a bubble sheet? Can you give me a blank Scantron? And so what you'll be able to do is you can X out your questions, and you can say, okay, I'm on number 3. I know that A and C are not correct. It'll help you to kind of focus in on, okay, I need to narrow down to B and D. Kind of having you be able to write. Like, I'm a big thing. Yes, this video is going to be saved. So just ask if this can be saved. Yes, I will save it on YouTube.
I typed IGTV and it auto-corrected to guts.
To guts.
It's going to be saved to guts.
Yes, I will put it on my channel. On basically, you just search AP Bio Penguins on YouTube. You'll pop all of our videos, including the videos we made last year in our 3-part process of one night. you will also find the videos for Marco Learning on my website. So apbiopenguins.weebly.com. And I've put everything up there, plus anything I do with Melanie is up there. There's a link to Melanie's website. If you go to my website, there's a little APsolute RecAP, and you click on it, it takes you right to her page. And it's perfect because it goes right to the podcast page.
Ah,
you can actually start listening to the podcast from her page, which is great. So there's some more questions that popped up in the things. Let's see if we have any that are on topic. Tips for FRQs, they kill me.
They kill you. Passwords. Passwords. Don't talk about things that aren't related to the question. Just because it's a vocab word, don't define it necessarily because the reader knows the word too. And that won't earn you points.
Yeah.
Those are my tips.
Okay. Let me pop this up there. And let me give a shout out. Hi, Elizabeth. This is
do you have a student
biological
in here?
Penguin. It's a biological penguin.
Biological penguin, a real one in the flesh.
It's a real, well, they're all penguins, but I mean, like, every single one of you are penguins. But, like, this is a student that's in my actual classroom.
Oh, my God.
So they want to know the difference between photosystem 1 and photosystem 2. So we already mentioned photosystem 2 is going to be the first one you see, and photosystem 1 is the second one. And I believe they call the first one P680 and the second one P700. And it's because they absorb slightly different wavelengths. Overall, they're basically the same. They're going to have a reaction center complex in the middle, which is made of that chlorophyll A. And then they're going to have these accessory pigments that have the job to absorb the solar radiation, the photons. And they get excited, sending their electron up to a higher energy level. And then when the electron pops down, it passes that energy to the next one. I kind of think of it like the wave when you're like at a sporting arrangement. And you're kind of, next one. Yes. And so they both have that, gets that reaction center complex. The reaction center complex gets oxidized by the primary electron acceptor that goes down electron transport chain. This happens in both of them. The only main difference is which one happens first. Photosystem 2 happens in the linear chain first, and then specifically which wavelength. P680 would be photosystem 2, and P700, if I remember correctly, is photosystem 1.
Yeah, and you might get a graph referencing absorption of different wavelengths of light with some analytical questions referencing the electron transport chain. So not all.
Go, keep going with your question.
No, I was just saying not all plants absorb all wavelengths equally. What was your question?
Somebody said, is there a link to the podcast? Like, is it a quick, easy way to find it?
Yeah, gotcha. I think so.
Somebody just asked that question.
I don't know if you're going to be able to click on it in here, but I will put it.
Oh, good connection, Kaufman. I know this stuff, but I forget to say some stuff. Kaufman said, okay, let's make sure that we know about photosystem 2 is where photolysis occurs. Great job. Yes, that is where, because it's the first step, that's where the electron is regenerated from it losing some primary electron acceptor versus our photosystem 1 gets regenerated from the electron transport chain coming down. So we don't have photolysis occurring at photosystem 1. It only occurs at photosystem 2.
Right.
And so that link, if you're looking for it, is theapsolute.com/biology.
That's not even correct. Oh my goodness. It's theapsoluterecap.com. No, I forgot the last word. Theapsoluterecap.com. I don't think you can click on it anyways in here. But if you click my my Instagram handle, it's the first link in our bio. And honestly, you're welcome to come to our website, but just if you Google it, it pops right up. If you're a Spotify user or a Apple podcast user or a Google, wherever you podcast, it'll be there.
Okay, let's see if there's any more biology questions up in here. That's the same things we've already mentioned. So this is a quick one. FADH2, is it present in both photosynthesis and cellular respiration? Nope. It's only going to be found in cellular respiration. We only see it, actually, the only place we see it is in the Krebs cycle.
Yeah, and the thing not to fall into trap with is that whereas you're going to see NAD plus, is you're not going to see FADH plus. So it's FADH2 and then FADH as it's kind of like alternate forms.
Let's see. There's something just popped up in the chat. Like something just popped up in the question. I wish you would put them in order for me. Like I would know like which was what's the first one, the end one.
Yeah.
So we're talking about energy, how does the ATP ADP cycle work? Where does it get its energy from and how does the process occur?
That was a question that popped up?
Yes.
Okay, yeah. So it is glitching for me because I'm not seeing anything. It's all
Oh, really?
I see the first question that you posted way back when we started, and it's never changed.
Oh,
Is it glitching for any of
wow.
you guys? Are you able to see new questions? I'm just curious. Um,
So ATP is adenosine triphosphate. ADP is adenosine diphosphate.
Diphosphate.
So when we go through a exergonic reaction, so I don't know, when the protons slow down their concentration gradient, that would be exergonic, so releasing energy. That potential energy is getting released. Thank you, Elizabeth. She says she sees new questions. It's just you. Hey, but at least you're still with me.
Yeah, I'm using an iPad today, so we're learning.
So that exergonic reaction provides the energy for the endergonic reaction of adding a phosphate to ADP. So all we see with the ATP ADP cycle is whether we're gaining or losing a phosphate. If we attach the phosphate, it means that we are absorbing that energy to make that bond. And then whenever we go ATP to ADP, we release that phosphate. Usually we phosphorylate something. I always tell my students, you phosphorylate, it's primed to do work.
Yeah.
How does ATP synthase make ATP? How does ATP synthase make ATP? It looks like a rotor almost.
It does,
like
yeah. So your
binds to
hydrogens
it.
are flowing through it. Your little rotor's turning. Yeah.
Yeah, and it's kind of like a turbine. Like it turns it, and as we said before, when you bind something to a protein, it changes shape. That changes shape conformation. I think it takes like 3 protons to move
three
it.
turns, I think, yeah.
In order to get it to have enough energy released from the phosphate moving, not phosphate, from the proton moving down its concentration gradient, that provides the energy to attach that phosphate.
Yeah, and so like a water wheel, right? Potential energy of water falling through a water wheel would work really similarly. So high to low, right? Proton motive force, awesome. High to low concentration flow here through a proton.
So it is 8:57.
It is, yeah.
So I'm going to say, last tips before we leave. Don't get overwhelmed when you look at Unit 3 stuff. There is a lot of steps for glycolysis, a lot of steps for Krebs cycle. There's a lot going on in these. Just make sure that you kind of take a deep breath. You've got this, especially when you see it for response. If you see any questions on this, just take a breather. You have this. I believe in you. You're a penguin and you're dressed with success. Anything you want to say?
Yeah, make some graphic organizers to calm yourself. That way you can pull in what you need to and kind of like leave on the periphery the things that you don't need to overwhelm yourself with, right? So put it all that's most important in front of you because we're getting down to crunch time. It's 2 months away and we need to start to prioritize the big picture at times right now.
And if you liked this live, we are going to go live again on Unit 4 and some of 5.
Some of 5,
which will be on March 21st.
Yeah, that sounds right.
And then I also will have a live with Marco Learning on the 20th, right before it. So if you're liking these live sessions, definitely, come see us again on March 21st, because we will do this crazy fun thing again if you think that we're fun to watch, because I think we're fun to watch.
we get a little weird with it. That's kind of part of the process, right? You can't study science and not be a little weird.
Of course not. We have to, you had to have fun with it, right? Because if we were just straight content, that would just be boring.
No. So if you're looking for the podcast, we release new biology episodes every Wednesday. We also have episodes in chemistry, government, music theory, and physics 1. And we will be hosting the binge, which is the biology binge review classes starting in April. So you can find links to that on our website if you're interested. They won't be held on Instagram. They'll be a little bit more concentrated with a smaller group of students for review.
So I hope everyone has a wonderful night. Thank you, Melanie, for coming in and doing this with me. And I hope you get all of your lesson planning done. And I don't know when I'm going to get mine done.
Me too. Have a great week, everybody.
Good night. Bye, penguins. Bye, recappers. And have a great night. Bye. Wrong button.